Evo 2, the largest open-source AI model for biology. Introduction. On February 19, 2025, the ARC Institute unveiled Evo 2, the largest open-source AI model for biology to date. Developed in collaboration with NVIDIA, Stanford University, UC Berkeley, and UC San Francisco, this transformative AI is trained on a staggering 9.3 trillion nucleotides derived from over 128,000 genomes from various lifeforms including bacteria, humans, and plants. Evo 2 possesses the capability to predict which mutations in genes are likely to be pathogenic and can even design entire eukaryotic genomes, marking a significant advancement in the field of generative biology. Key features and capabilities. 1. Disease prediction and genome design. Evo 2 excels at identifying mutations in human genes that may lead to diseases, achieving over 90% accuracy in distinguishing benign from potentially pathogenic mutations such as those in the BRCA1 gene linked to breast cancer. Furthermore, it can generate long genetic sequences, simulating evolutionary processes to produce novel gene designs that could potentially be utilized in gene therapies. 2. Technical innovation. The model is built on a unique architecture known as StripedHyena 2, allowing it to process up to 1 million nucleotides simultaneously. This advancement enables the discovery of long-distance interactions between genes, significantly enhancing data handling capabilities compared to its predecessor Evo 1, which focused on single-cell genomes. 3. Open-source access. The complete codebase, including training data, model weights, and a user-friendly interface dubbed Evo Designer, is publicly available on ARC Institute's GitHub page. This initiative aims to democratize access to advanced AI tools in biological research, fostering collaboration and innovation across the scientific community. For ethical considerations, to mitigate potential risks, Evo 2 was designed without including genomes of pathogens that infect humans or complex organisms, ensuring that the model cannot provide actionable insights regarding dangerous viruses or bacteria. Development and collaboration. Evo 2's development is a product of extensive collaboration among leading institutions. The ARC Institute, a non-profit research organization focused on accelerating scientific progress, spearheaded the project, partnering with NVIDIA, which provided the necessary AI computing infrastructure, and academic institutions such as Stanford, UC Berkeley, and UC San Francisco. The team combined expertise from various fields including bioengineering, chemical engineering, and computational biology. The model was trained on the NVIDIA DGX Cloud AI platform via AWS, utilizing over 2,000 NVIDIA H100 GPUs. This hardware was crucial to handle the immense data scale, allowing Evo 2 to process genetic sequences much larger than previous models. Greg Brockman, co-founder and president of OpenAI, contributed to the technical development during his sabbatical, leading the creation of the StripedHyena 2 architecture, which enables faster and more efficient data processing. Implications for biological research. Evo 2 is poised to revolutionize drug discovery and genetic engineering. By accurately identifying genetic mutations associated with diseases, it can significantly reduce the time and resources needed for experimental research. For instance, its high accuracy in predicting the pathogenicity of mutations could streamline the process of understanding genetic diseases, potentially leading to faster development of targeted therapies. Moreover, Evo 2's ability to design new genomes opens up possibilities for creating synthetic biological tools and treatments. For instance, researchers could engineer genetic elements that activate only in specific cell types, enhancing the precision of gene therapies and minimizing side effects. The future of Evo 2 and generative biology. The Evo team envisions that Evo 2 will serve as a foundational model for developing more specialized AI tools in biology. By providing a robust platform, researchers can build applications on top of Evo 2, from predicting the impact of specific DNA mutations on protein function to designing genetic sequences with tailored functionalities. The developers also emphasize the importance of ethical considerations in deploying such powerful technology. By excluding pathogen genomes from its training data, the team aims to prevent misuse and ensure that Evo 2 contributes positively to scientific advancement. Conclusion: Evo 2 represents a groundbreaking achievement in the integration of artificial intelligence and biology. Its capacity to predict genetic mutations, design novel genomes, and democratize access to advanced AI tools positions it as a pivotal resource for researchers worldwide. As the scientific community begins to harness the capabilities of Evo 2, the potential for accelerating discovery and innovation in understanding genetic diseases and advancing biotechnological applications is immense. The collaborative efforts that brought Evo 2 to fruition may well set a new standard for the future of AI applications in the life sciences. Evo 2 is a groundbreaking AI model for biological research developed through a collaboration among several prestigious institutions, including the ARC Institute, NVIDIA, Stanford University, UC Berkeley, and UC San Francisco. Unveiled on February 19, 2025, this model is notable for its unprecedented scale, being trained on an extensive dataset of 9.3 trillion nucleotides derived from over 128,000 genomes across a wide variety of life forms. This capability allows Evo 2 to predict pathogenic mutations in genes and even design entire eukaryotic genomes, marking a significant advancement in the field of generative biology. One of the most counterintuitive aspects of Evo 2 is its open-source nature. While AI models, particularly large ones, are often proprietary and closely guarded, Evo 2 is fully accessible on GitHub. This democratization of advanced AI tools stands in contrast to the trend of restricting access to powerful technologies, suggesting a belief that collaboration and transparency can drive innovation in scientific research. By making Evo 2 open source, the developers aim to foster a global research community that can build upon and refine the model's capabilities, potentially leading to unexpected applications and discoveries that would not be possible in a closed environment. Moreover, Evo 2's ability to predict pathogenic mutations with over 90% accuracy, particularly in genes like BRCA1, underscores its potential to revolutionize medical research. The model's predictions can significantly reduce the time and resources traditionally required for laboratory experiments, a shift that could accelerate drug discovery and the development of genetic therapies. This efficiency might seem counterintuitive in a field often characterized by painstaking and time-consuming research processes. The prospect of quickly identifying disease-causing mutations may lead to a rethinking of how genetic research is conducted, allowing scientists to focus on more targeted and potentially impactful studies. The technical innovation behind Evo 2, specifically the architecture known as StripedHyena 2, is another area of counterintuitive advancement. This architecture enables Evo 2 to process up to 1 million nucleotides at once, allowing for the detection of long-distance interactions between genes. This capability challenges traditional approaches in genetic research, which often focus on shorter sequences and localized interactions. By expanding the context within which genetic relationships are examined, Evo 2 opens the door to new insights into how genes influence one another across vast genomic distances, leading to a more holistic understanding of genetic function and interaction. Interestingly, the ethical considerations surrounding Evo 2's development further highlight a counterintuitive approach. The developers intentionally excluded genomes of pathogens that infect humans and complex organisms from the training dataset to mitigate potential risks. This decision contrasts with the common practice in AI, where datasets are often expanded to include as much information as possible regardless of potential safety concerns. By prioritizing ethical considerations over sheer data volume, the creators of Evo 2 set a precedent for responsible AI development, emphasizing the need for safety and ethical standards in the rapidly evolving field of biotechnology. Another notable feature of Evo 2 is its potential for generating new genetic sequences. Unlike traditional evolutionary processes, which are random and time-consuming, Evo 2 can speed up evolution by predicting and designing genetic sequences that could lead to beneficial traits or functions. This capability could radically change the landscape of genetic engineering and synthetic biology, challenging the notion that evolutionary processes must be slow and natural. Instead, Evo 2 presents a model where targeted engineered changes can be made to DNA at unprecedented speeds, pushing the boundaries of what is possible in genetic modification and therapeutic development. Furthermore, the collaborative nature of Evo 2's development is worth noting. The project required the integration of expertise from various fields, including machine learning, biology, and experimental research. This interdisciplinary approach is increasingly recognized as essential in solving complex scientific challenges, yet it remains relatively rare in practice. The successful collaboration among diverse institutions exemplifies how breaking down silos in scientific research can lead to significant advancements. A realization that may seem counterintuitive in a competitive academic environment. In conclusion, Evo 2 represents a transformative advancement in biology, offering a range of counterintuitive possibilities that challenge traditional methodologies and ethical frameworks in scientific research. Its open-source nature, predictive capabilities, innovative architecture, and ethical considerations collectively suggest a shift towards a more collaborative, efficient, and responsible approach to biological research. The implications of Evo 2 extend beyond mere technological advancement. They invite a rethinking of how science can be conducted in an era increasingly defined by artificial intelligence and collaborative innovation. Evo 2 is a groundbreaking achievement in the realm of artificial intelligence and biology, representing a shift in how we understand and manipulate genetic information across various life forms. While much of the discourse surrounding Evo 2 highlights its technical capabilities and potential applications, there are several nuanced aspects of this model that are likely to be overlooked yet hold significant importance for the future of biological research in medicine. 1. The scale of training data and its implications. One of the most impressive features of Evo 2 is its training on an unprecedented dataset of 9.3 trillion nucleotides from over 128,000 genomes, encompassing a wide range of lifeforms from bacteria to humans. Most discussions emphasize the sheer size of this dataset, but the implications of such a comprehensive training foundation often go unexamined. Significance. The diversity of the training data allows Evo 2 to identify evolutionary patterns that might be invisible in smaller or less diverse datasets. This broad genetic library enables the model to discern relationships and predict mutations across different species, potentially leading to discoveries that can be applied to various fields, such as personalized medicine or synthetic biology. The ability to leverage a more generalized understanding of biology could also pave the way for more robust and versatile AI applications in genetics. 2. Long-distance gene interactions. Evo 2's capability to process up to 1 million nucleotides at once is a technical feat that deserves deeper exploration. While this aspect is often presented as a technical specification, its implications for understanding gene interactions are profound. Significance. Traditional genetic analysis typically focuses on shorter sequences, which can overlook the significance of long-distance regulatory interactions between genes that may be physically separated within the DNA strand. By analyzing larger segments of DNA, Evo 2 can uncover intricate networks of gene regulation, thereby offering insights into complex traits and diseases that are influenced by multiple genes. This could lead to a paradigm shift in how researchers approach genetic disorders and their treatment, moving from a reductionist view to a more holistic understanding of genomic interactions. 3. Generative biology and its ethical dimensions. Evo 2's ability to design new eukaryotic genomes and predict the pathogenicity of mutations raises ethical questions that are often glossed over in discussions about its capabilities. The model's generative power could allow researchers to create novel genetic sequences that do not exist in nature, pushing the boundaries of genetic engineering. Significance. The ethical implications of creating new forms of life or modifying existing organisms are vast. While the exclusion of pathogenic genomes from the training set is a necessary precaution, The potential misuse of generative biotechnology in creating harmful organisms or altering ecosystems cannot be ignored. The scientific community must engage in ongoing conversations about the ethical boundaries of synthetic biology and ensure that such powerful tools are used responsibly and for the benefit of humanity. This includes establishing guidelines and frameworks to govern the applications of Evo 2 and similar technologies. 4. Open source advantage. The decision to make Evo 2 fully open source has been highlighted as a democratizing force in scientific research. However, the broader impact of this choice on collaboration and innovation within the scientific community is often undervalued. Significance: open source models encourage a culture of transparency and collaboration among researchers from diverse backgrounds and institutions. By allowing global access to Evo 2's capabilities, the ARC Institute is fostering an environment where innovative applications can emerge from unexpected places. This could lead to accelerated discoveries in various domains. As researchers can build upon each other's work without the barriers typically associated with proprietary technologies. Moreover, it establishes a precedent for future AI developments in biological research, potentially leading to a more collaborative and less siloed approach to scientific inquiry. 5. Potential for specialized models. Evo 2 is described as a foundational model that could support the development of more specialized AI applications. This aspect is critical as it indicates a shift from one-size-fits-all solutions to tailored approaches in biological research. Significance: the ability to create specialized models based on Evo 2's architecture could enhance research in specific areas such as oncology, rare genetic disorders, or agricultural biotechnology. By refining the model for particular applications, researchers can generate more precise predictions and designs, ultimately leading to more effective solutions in their respective fields. This modular approach to AI in biology could revolutionize the way researchers tailor their tools to meet specific challenges, making the research process more efficient and impactful. 6. Impact on drug discovery and development. While Evo 2's capabilities in predicting pathogenic mutations and designing genomes are often highlighted, the implications for drug discovery are potentially transformative but less emphasized. Significance: by accurately predicting which genetic mutations are benign and which are pathogenic, Evo 2 could significantly reduce the time and cost associated with drug development processes. Traditionally, identifying the genetic basis of diseases requires extensive laboratory work and animal models, which can take years. Evo 2's predictive power allows researchers to focus on the most promising genetic targets, thereby accelerating the pipeline from discovery to therapeutic application. This could lead to faster, more efficient treatments for diseases, ultimately benefiting patients and healthcare systems alike. Conclusion: Evo 2 represents a significant leap in our ability to understand and manipulate genetic information. While its capabilities are often discussed in terms of technical specifications and potential applications, the broader implications of its architecture, data diversity, ethical considerations, collaborative potential, and impact on drug discovery deserve careful consideration. As the scientific community begins to harness the power of Evo 2, these overlooked aspects could prove to be as crucial as the technology itself in shaping the future of biology and medicine. 1. Disease prediction and genome design. Evo 2's ability to identify pathogenic mutations with over 90% accuracy, particularly in genes like BRCA1 associated with breast cancer, represents a monumental leap in genetic research. This capability not only aids in understanding genetic diseases but also accelerates drug development by potentially reducing the need for extensive cell or animal studies. The model's proficiency in generating new gene sequences means it could also pave the way for innovative gene therapies tailored to specific conditions, enhancing the precision of medical interventions. 2. Technical innovations and scalability. The underlying architecture of Evo 2, known as StripedHyena 2, marks a significant advancement in AI modeling for biology. It processes up to 1 million nucleotides simultaneously, enabling the analysis of long-distance gene interactions that were previously difficult to study. This technical leap allows Evo2 to utilize a dataset of 9.3 trillion nucleotides, 30 times more than its predecessor. Evo 1. Such scalability not only enhances the model's predictive capabilities but also sets a precedent for future AI applications in biological research. 3. Open source accessibility. The decision to make Evo2 fully open source by providing access to its code model weights and training data democratizes advanced AI tools in biological research. This collaborative approach is significant as it fosters innovation and enables researchers worldwide to leverage Evo2 for their studies. By allowing scientists to build upon this foundational model, Evo2 can catalyze new discoveries and applications in genetics, bioengineering, and medicine. For ethical considerations and safety measures, Evo2 was designed with ethical considerations in mind. Specifically by excluding genomes of human pathogens and ensuring the model does not yield actionable insights regarding dangerous viruses or bacteria. This thoughtful approach to model training and development not only mitigates potential misuse but also establishes a framework for responsible AI deployment in sensitive areas like genetics and biotechnology. The commitment to ethical practices may serve as a guideline for future AI developments in the life sciences, balancing innovation with safety. 1. The Human Genome Project (HGP). Initiated in 1990 and completed in 2003, the Human Genome Project was an international scientific research project aimed at mapping and understanding all the genes of the human species. It was one of the largest collaborative biological projects ever undertaken, involving scientists from around the world. The project successfully sequenced the entire human genome, which consists of over 3 billion nucleotides. This monumental achievement has paved the way for numerous advancements in genetics, medicine, and biotechnology. Similar to how Evo2 aims to transform the field of biology through AI-driven genome analysis and design. 2. CRISPR-Cas9 gene editing. Discovered in the early 2010s, CRISPR-Cas9 is a revolutionary gene editing technology that allows for precise modifications to DNA. This tool has been utilized to edit genes in various organisms, including humans, plants, and animals. The ability to target specific genetic sequences and alter them with high precision has opened new avenues for research and therapeutic applications, much like the capabilities of Evo2 in designing new genomic sequences and predicting pathogenic mutations. Both CRISPR and Evo2 represent significant leaps forward in our ability to manipulate genetic information. 3. Artificial intelligence in drug discovery. In recent years, the application of AI in drug discovery has gained prominence. With platforms like Atomwise and Benevolent AI utilizing machine learning algorithms to analyze vast datasets and predict how different compounds will interact with biological targets. These AI systems can significantly speed up the drug discovery process, much like Evo2's ability to predict disease-causing mutations and generate genetic sequences. Both demonstrate the powerful intersection of AI and biology, pushing the boundaries of what is possible in research and development. Future outlook for Evo2, predictions and implications. As Evo2 emerges as the largest open-source AI model in biology, its capabilities and implications for the future of genetic research and biotechnology are profound. The model, which can accurately predict pathogenic mutations and design new eukaryotic genomes, is poised to revolutionize the fields of medicine, environmental science, and agricultural biotechnology. Here are several predictions regarding its future impact. 1. Accelerated drug discovery and development. Evo2's ability to identify mutations with pathogenic potential will significantly expedite the drug discovery process. Researchers will rely on Evo2 to pinpoint genetic variations linked to diseases, allowing for targeted drug development. This efficiency could reduce the time and cost associated with bringing new therapies to market, potentially leading to more personalized medicine approaches that cater to individual genetic profiles. 2. Advancements in gene therapy. The model's genome design capabilities will enable the creation of tailored gene therapies that specifically target cells affected by diseases, minimizing side effects. As more researchers adopt Evo2 for designing precise genetic elements, we can anticipate breakthroughs in treating genetic disorders, cancer, and other complex diseases. The ability to engineer genetic sequences could also facilitate the development of smart therapies that activate only in certain tissues. 3. Transforming synthetic biology. Evo2 is likely to become a foundational tool in synthetic biology, enabling the design of novel biological tools and organisms. This could lead to the creation of microorganisms engineered for specific tasks such as bioremediation of pollutants or bioproduction of sustainable materials. Researchers could leverage Evo2 to simulate evolutionary processes, leading to the discovery of new enzymes or metabolic pathways that enhance industrial biotechnology applications. 4. Enhanced understanding of evolutionary biology. By using Evo2 to analyze vast datasets of genetic information across diverse species, scientists will gain deeper insights into evolutionary patterns and mechanisms. This could refine our understanding of how genetic mutations contribute to adaptation and speciation, potentially reshaping evolutionary theory itself. As researchers explore these connections, Evo may second also reveal previously unknown relationships between genes and traits. 5. Global collaboration in open science. The open-source nature of Evo2 encourages widespread collaboration among researchers, fostering an environment of shared knowledge and innovation. This collaborative spirit is likely to lead to rapid advancements in the field as scientists from various disciplines work together to harness the model's capabilities. We might see the rise of a global community focused on tackling complex biological challenges, democratizing access to cutting-edge technology. 6. Ethical and safety considerations. As Evo2 gains traction, ethical considerations will come to the forefront. While the model has been designed with safeguards against misuse, ongoing discussions regarding bioethics, genetic privacy, and the implications of genome editing will intensify. Researchers and policymakers will need to establish guidelines that ensure responsible use of this technology. Balancing innovation with safety and ethical responsibility. 7. Integration with other AI and biological models. The architecture of Evo may second inspire the development of specialized AI models tailored to specific biological questions, similar to how Evo2 serves as a foundational kernel. We can expect to see integration with other computational models that analyze complex biological systems, enhancing our understanding of gene interactions, signaling pathways, and cellular behavior. This synergistic approach could lead to breakthroughs in systems biology. 8. Impact on education and training. As Evo2 becomes a standard tool in biological research, educational institutions will likely integrate its use into curricula. Students and emerging scientists will gain hands-on experience with advanced AI tools, equipping them with the skills needed to navigate the future of biotechnology. This shift could inspire a new generation of researchers adept at combining biological sciences with computational methods. 9. Environmental applications. Evo2's ability to design genomes could be harnessed for environmental conservation efforts. For example, researchers could engineer plants or microorganisms to enhance carbon capture, resist climate stressors, or degrade environmental pollutants. This application could play a vital role in addressing climate change and promoting sustainable practices in agriculture and industry. 10. Long-term vision for artificial life. Looking further ahead, Evo may second contribute to the development of artificial life forms designed for specific functions. This could lead to advancements in biotechnology where engineered organisms perform tailored tasks such as producing biofuels or pharmaceuticals. The exploration of synthetic organisms could redefine our relationship with biology and the natural world. In summary, the future of Evo2 is rich with possibilities that extend across multiple domains of science and technology. Its potential to transform drug discovery, enhance our understanding of genetics, and foster global collaboration exemplifies the groundbreaking impact of AI in biology. As researchers continue to explore its capabilities, the implications for health industry and the environment will unfold, shaping the future of life sciences in unprecedented ways.